Oh, hopefully you heard that. We're actually recording meetings so people can listen to it later and get the information. So again, my name is Dave Block, and this is our second office hour with Dave and Anita. In this office hour, we're going to focus on an important problem that vineyards in California have been facing for a number of years now, and that's the problem of red blotch disease. Recently, a group of researchers here at UC Davis and in other parts of the country received a large specialty crop research initiative, or SCRI, grant from the USDA to work on red blotch and to figure out the causes and also some of the ways to get around the problem or to solve the problem. And the project leader for that group is Anita Oberholster. So I want to actually take a moment to introduce Anita, and then she'll introduce the project and some of the other researchers working on it who are also on the call. They'll give a short presentation on the research that they're just starting now or have already been doing in some cases. And then after that, we'll just open it up to questions. If you're new to Zoom, and I would assume at this point, a lot of you who've been using Zoom for a lot in the last month or so, but if you are new to Zoom or large groups of Zoom people, in your participants panel that you can open up at the bottom of your screen, it ends up on the right side. There's a little blue hand that you can click on to raise your hand to ask a question, or you can write something in chat if you want, and either I can read it to the group, or Karen Block, who's also on the call, can read it to the group as well. So that's just some mechanics. So with that, I'd like to introduce Anita Oberholster, who's the project manager for the SCRI grant. Anita has degrees in chemistry and wine chemistry, undergraduate from University of Stellenbosch, and PhD from University of Adelaide and AWRI in wine chemistry. After that, she went to work at the University of Stellenbosch in South Africa for a number of years before joining us in 2011 as cooperative extension specialist in Enology. So she's been with us for 9 years, and not to embarrass her, but in those 9 years, she's made a lot of connections through the industry, and for the last 2 years has been named by Wine Business Monthly as one of the top 50 most influential people in the industry in the US. So with that, I'm going to turn it over to Anita to introduce the topic and her team.
Well, that was a very formal introduction. I was going to go with, hey, that's Anita. She's at the department. So I'm supposed to share my screen. So I'm quickly going to do that so that you have, I have too many screens so that you can see that. Did that work?
Yep, that worked.
Okay, my view just changed. Let me just try and figure out what changed here. It doesn't help when you're working with 3 screens. It seems to be going everywhere. Okay, so I'm going to look here, but my presentation is actually there. So basically, wait, I'm going to try and move it quickly.
Anita, you can probably go to full screen to presentation mode.
Okay. Okay, now I can see it. That helps. Thank you very much. Okay. So
it didn't change on our screens though.
Yeah, you probably have 2 screens.
I do.
Yeah. So I'll move it to the presentation screen probably.
If not, I think this will work, Anita.
Yeah, that's fine.
It's fine. All I'm doing.
Well, we do want to see your slides as you go, though.
Screen. Okay, there we go.
That's better.
There's technology to worry about. As Dave said, I've been working on grapevine red blotch virus. And on this call, we also have some of my collaborators. Con Cottrell, who's the Cooperative Extension Specialist in Viticulture, based here at the department, as well as at the Oakville Experimental Station, as well as Sudhi Sudarshana. He is a research biologist of the USDA ARS. He's based at the Department of Plant Pathology here at UC Davis. And Emeritus Professor Frank Zalem, he was at the Department of Entomology and Mythology, and was also an extension specialist. So obviously we're not the only people working on grapevine red blotch virus, but I thought that between a chemist, a viticulturist, an entomologist, and a virologist, we could potentially answer most of your questions. And it makes the group just a little bit more manageable and smaller. So first up, it's going to be a very, very brief introduction. The idea is really more to focus on questions and answers. For those of you that don't know that much about grapevine red blotch virus, perhaps because you wanted to watch something other than CNN, no. So grapevine red blotch virus is the causal agent of grapevine red blotch disease. And the characteristic symptoms of this disease for red grape cultivars are these red blotches on the leaf blades and on the margins. You also get the reddening of veins, of secondary and tertiary veins on the leaf undersides, no rolling. So this is a major difference from leaf roll, that the leaf don't roll. You also get them very similar to leaf roll, that the leaves turn red in the early fall, and those closest to the base of the shoots are the most severely infected. To see the symptoms in a white grape variety is a little bit more difficult. You can find marginal necrosis, like you can see in the photo provided by Rhonda Smith. And the symptoms can seem similar to a severe late season potassium deficiency. What we've been able to consistently find in the last few years are the impact of this disease on grape composition is a decrease in total soluble solids, a decrease in anthocyanin content in red grapes, and a decrease in yield. However, disease expression does depend on genotypic and environmental factors. I say this because we've been looking at different varieties, sometimes on different rootstocks at different sites over multiple seasons. And what we found is that definitely there's different impacts in different varieties, but within the same variety, you see differing impacts depending on the site, depending on the rootstock. Also, we see significant impact due to the season, which probably indicates that there's a synergistic impact of climate, either nutrient status, either water or temperature. The latter definitely has an influence. To give you a brief timeline here. So in 2012, they developed a qPCR test that could test for the grapevine red blotch virus. So we could determine, yes, this is definitely the causal agent of grapevine red blotch disease. They also sequenced the genome of the virus, finding that it's a single-stranded DNA virus, also known as a Gemini virus. Work that Sudhi's done in collaboration with other colleagues found that you have sugar accumulation in the leaves as well as a buildup of amino acids. Frank Zalem's group found actually a potential vector, the three-cornered alfalfa leafhopper, also Spasticillus festinus. I probably now just butchered that name if you want to be scientific. In the meantime, Dr. Cantu did work looking at the impact on transcriptional factors and hormone networks and found that there is a delay in the metabolic pathway, specifically the phenylpropanoid pathway. And that is the pathway that is responsible for the synthesis of anthocyanins as well as other important phenolics. Both Kine and myself has done work looking at the impact on plant physiology, and I've looked more at grape composition and wine composition. Kine has found that the virus impedes carbon translocation mechanisms, leading to impaired ripening. I've observed the result of that in grape composition, like I've mentioned, both delays in sugar content, anthocyanin synthesis. But we've also noticed a trend of an increase in tannin synthesis in the skins. This may be in a response as a defense mechanism response to the virus itself. We've also noticed an impact on the volatile composition of the grapes. So the impact isn't just on phenolics, there's also other compounds that are impacted, both primary and secondary metabolites. This is a slide that was provided by Khan, and he can address this in more detail during the question and answer session, but I'm going to do my best to summarize this. So grapevine red blotch disease impairs sugar transport. This results in carbohydrate accumulation in the leaves. So it's not moving from the leaves to the berries. Due to this accumulation in the leaves, it activates anthocyanin synthesis in the leaves, causing the leaves to turn red. In the meantime, because you have slower or less carbohydrate accumulation in the berries, it is impeding those same ripening processes. So everything happens slower and you don't get that activation of anthocyanins as early as you normally would. Also, you get stomatal closure, reduced transpiration, which results in actually a better water status in the plant, causing bigger berries, diluting berry content. So that basically exasperates the problems of less phenolics and less color and less sugar. That's the short-term impact. The long-term impact is because you have grapevine red blotch disease impairing sugar transport and having later ripening, you have lower capacity to accumulate reserves in the plants, weakening the plant, reducing bud fruitfulness and vigor. Well, that's what we suppose. And that's definitely what we need to study further. Now, as Dave has mentioned, we were successful after 3 long years of trying to obtain this USDA NIFA Specialty Crop Research Initiative Standard Research Extension Project. That's what that acronym stands for. It's a 4-year project. We have 12 principal investigators and an additional 10 collaborators. We're all based in California and Oregon State. And you may ask, why California and Oregon State? With feedback with stakeholders as well, we've determined that those are the 2 states that have the most spread of this virus, other than through propagation, that shows a vector spread. And that's one of the major focuses of this grant. Also, by spreading ourselves not across the country and very thinly, we can really investigate the problems more deeply, doing almost a full life cycle of this disease. Basically, more bang for your buck because it's not spreading among 36 investigators, but only 12 at this stage. Now, this is a logic model. This is something we need to do when we apply for grants. This was mostly Sudi's hard work, and you really don't have to read all this detail. I'm going to summarize it for you. But I just want to show you that this grant is much more complex than what I'm going to distill it down to. And basically, in a logic model, you need to show the flow of activities. You can see there's 3 major objectives with sub-objectives and the hypotheses that they are based on, that they're going to evaluate and test, the major outputs that we expect from each objective, as well as then the short, midterm, and long-term outputs or outcomes expected from the whole grant. To summarize the objectives, we're going to determine the prevalence and spread of grapevine red blotch virus in California and Oregon State, identify alternative hosts and reservoirs, determine the biology of potential vectors, virus acquisition and transmission, evaluate viticulture practices to mitigate, determine impact on grape and wine quality, develop cost-effective diagnostics tools, assess economic impact, and develop sustainable integrated pest management strategies. Now that is quite a lot. And we're going to hope to make a lot of progress in each of these objectives. Many times that happens with many grants. So you make progress, you create more questions, and you have a follow-up grant to address the new questions that arose while you were studying these problems. But this is what we're aiming to do. And in these objectives, Khan and Frank and Sudi and myself can definitely address most of the questions if you have questions regarding this and other things you think we should be including. But obviously we're not the only investigators. Like I said, we have 12 investigators. principal investigators and then another 10 collaborators on this grant. And we're all also doing other research focus on red blotch disease on the side. And obviously there's additional researchers in the country that's working on this problem. Well, that is actually my short, hopefully not too long spiel. And now we can open it up for questions, but I think Dave is gonna start off the questioning, and as he asks questions, if you have anything that you would like to add, please chip in.
Yeah, so I have a couple questions to just start things off, but if while someone's answering the questions, you think of something, again, you can click the raise hand button in the participants panel or put something in chat. Or otherwise get my attention, and we'll be happy to get your question on. So this is a question for Frank as a first question. I wonder if you can address if there are any confirmed vectors for the virus in the field, and how do you find potential vectors?
Okay, that's a very good question. Anita mentioned in her presentation that we did find transmission of the virus in a lab and greenhouse study, which was published in 2016. And that insect that we found as a potential vector was the three-cornered alfalfa hopper, which is a tree hopper. It's a membracid. And we found that basically we've been trying to repeat that transmission study in the field, and we've never been able to actually get transmission occurring in the field. And there has been no other insect that has been identified to transmit the virus in the field at this point. We believe that since this is a Gemini virus, it has to be insect transmitted, or it's likely to be insect transmitted. All Gemini viruses are transmitted by insects. Interestingly, the closest virus to grape red blotch virus is a tomato curly
pseudo curly top.
Thank you. Tomato pseudo curly top virus. And it's transmitted also by a tree hopper, and that's the only other virus that's transmitted by a tree hopper. So there's a lot of circumstantial evidence that we should be able to get transmission in the field. Now, I'll say that how we go about identifying a vector, which was the other part of your question. Basically, we try to identify vineyards where we can confirm spread has been occurring. And so we have several vineyards that we've followed for several years. And before I was involved, Sudi and others were monitoring spread in vineyards. We're able to document that. So we looked for potential vectors in those vineyards where spread was occurring. We look at very grossly, is the virus present in any of these potential vectors? And so we can look in the whole body and determine if there's a virus present or not. Then more recently, we've been able to dissect the salivary glands from insects. And so in order to transmit the insect, if it's a persistent virus, is likely to be present in the salivary glands as well. And so we look at the salivary glands as well from insects we collect in these vineyards. And if we have species that have the virus present in the bodies that are present commonly in the salivary glands as well, then those are insects we try to do transmission studies with. And so we for the most part, we take those, put them on a recipient plant so we know are virus free. And then we have to wait. And we wait for, in the initial study, we didn't actually see any transmission for about 5 or 6, or any presence of the virus in the plant for about 5 or 6 months. and that was under greenhouse conditions. We don't know how long it takes for the virus to be expressed in the field. And so we have studies that are, what, Sudhi, as long as 3 years already that we've been trying to monitor to determine if we're getting the virus in those vines, and we have not had that yet. So we are still looking for other potential vectors. And one of the things we're looking for is are there other biotypes of three-cornered alfalfa hopper? And so we're actually doing some sequencing work with the three-cornered alfalfa hoppers that we're collecting to try to determine if there's potentially another biotype present, like there is in a good example of another Gemini virus is Bemisia tabasi, that has a number of different biotypes that have been identified, only some of which transmit the tomato yellow leaf curl virus, which is the virus that is associated with it. We may have something like that with three-cornered alfalfa hopper as well. We don't know that. We're also looking at other tree hoppers as potential vectors, and we've actually collected them, set up transmission studies in the lab and in the field. We're also looking at a few other candidates that have, again, presence in the salivary glands, presence in the bodies, are found commonly in these vineyards where we see spread. And these include a couple of leafhopper species as well as a Succeed. Those are other families of insects that potentially could be transmitting. So that's a long answer to your question. It's everything that we're doing at present.
Great, thank you. I suppose if it were simple, you would have already found the vector, right? So
yeah, I thought it was going to be a lot simpler than this, but it's really difficult in grape plants because it takes a long time potentially for this to be expressed. And they go dormant in the wintertime. We don't know the time of the year that transmission actually occurs in the field. So those are all things that we're investigating right now.
Great, thank you. So I, not seeing any immediate questions right now, I'll ask Sudhi a question if I could. So I'm sure there are a lot of people here listening who are thinking to themselves, how do I keep grapevine red blotch virus out of my vineyard and to protect their vineyard as much as possible? And I was wondering if you could talk about ways of protecting your vineyard from GRBV.
I wish there was a simple and easy answer for this. I think most importantly, we have to source grape wines tested for the virus already, and if we can replant affected vineyards and rogue the vines and replant tested red blotch virus-free wines, that is a starting point at this stage. And the next thing would be whether there is a potential vector around, whether there was a spread happening in the past or in the newly planted blocks. This is something that can be monitored by the appearance of symptomatic wines, or suddenly you'll see the wines having low bricks. So once you suspect and test them out for the virus, that's your potential hotspot for reinfection. At this point, we don't know much about the vector or its biology, so we don't have any measures to control the vector yet as a guideline. So at this point, the best thing we can do is keep roguing the infected plants and replant. That's the only thing we can do at this stage.
Okay. Thank you. And I don't know that it was with red blotch, but I think there were studies done maybe in South Africa on roguing for other diseases that have proven to be effective. I don't know if any of you want to comment on that.
Roguing has been a practice for leaf roll viruses known for a long time. But we also know the vector precisely there. So there is option for vector management for leaf roll viruses. unfortunately, we don't have that luxury for the red blotch virus yet.
Thank you. Khan, I have a question for you. And I think Anita touched on this a little bit in one of her slides, but I wonder if you could just kind of summarize what the impact of the virus is on vine physiology.
Certainly. The disease reveals itself with a complex symptomology. So in our work at Oakville and around the state, we identified short-term and long-term responses on the grapevine physiology. So mainly what the disease does is it impairs sugar transport. Sugar is transported from the leaves to the other actively growing tissue, such as the roots, shoot tips, and the berries. But specifically in this case, in the short term, what we see is carbohydrates are accumulated in the leaves. So we knew that. But when the carbohydrates start accumulating in the leaves, we start seeing stomatal closure. And when stomatal closure happens, we end up with a reduced transpiration of the grapevine as a whole. When transpiration is reduced, we get further reduced carbon assimilation by the leaves. And then when we have a reduced carbon assimilation, what we have is sugar eliciting anthocyanins. That's what gives the leaves their botch, like the botches on the leaves. And therefore, carbohydrates are then not accumulated in the berries. They're somewhat stuck in the leaves. So in this case, since we have a stomatal closure and a reduced transpiration, we have accumulation of the water not used in the soil, and we have a lower contribution of total soluble solids accumulation. This ends up with a delayed harvest with a greater juice pH and a lower color accumulation in the berries. Overall, the plant has a better water status, meaning that it never actually goes into the classical water stress that we see in the summer. More often than not, this reveals itself with bigger berries, and then you have the dilution of berry content with lower total soluble solids. In the long term, which we have not been able to study in vineyards because they're removed after 2 or 3 years after the impacts are found. Over the long term, grapevine red blotch disease would impair sugar transport with late ripening. This results in a lower capacity of the plant to accumulate reserves, such as starches and other complex sugars in the perennial tissue. Well, more often than not, what we expect to see is plant weakening, a reduced growth in the form of our roots. Then what we hope to study with this experiment that we have now is reduced bud fruitfulness and vigor for years to come. So that's what we see in the long-term picture with this disease affecting physiology of the plant. So.
Great, thank you. So I know Kevin has a question, and then there's a question on chat that got sent to me. So Kevin, why don't you go ahead and unmute yourself and ask your question?
So yeah, my question was, are there any fertility or water management practices to help mitigate some of the poor fruit quality things we're having with red blotch? We don't really have the option of replanting a vineyard here, so we're looking at trying to mitigate, best we can.
So I can answer that, but as Khan is the person that's actually doing all the hard work and has done the trials, and he's on the call, I'm going to transfer this to Khan to answer.
Initially, we tried looking at ripening agents such as homologs of abscisic acid sprays. But this is before we knew about the impediment in carbon translocation transport. So it's not necessarily a lack of hormones for the berries not to ripen. So the two things that we're going to look at very closely is potassium sprays or potassium injections rather into the soil profile. And with irrigation management, we are looking at low and high water through a sustained deficit irrigation. In this case, for them to go into stress. That if we cannot put them into stress, because when we looked at with two different rootstocks with a different hydric potential, for example, with a 420A and a 110R, 420A was not going into a stress at all, what we would term stress at like a -12 bars, but a 110R was somewhat going into a stress. So in this case, we have a lot of opportunity to explore these options. So at the moment, I don't have an answer for you, but more often than not, this will have to do something with the carbohydrate metabolism mediated through potassium transport.
And something to add, I know the Southern Oregon Research Station has done some work where they,
Yes.
remember that, Con, where
Mhm.
basically they have their standard nutrient program for their vines, where they did nothing, normal amount, and double the amount.
Mhm.
I didn't see an impact, but it could be specific to those specific vines and what their natural health state is, and how much they get through the soils. And it
Yeah.
could be that just nothing and double was too extreme. I don't know. So we
We
went
saw some
further.
we saw some telltale signs of this in the year 2018 in Napa Valley and Sonoma County, because we use potassium sparingly in coastal areas, and with the high or a bumper crop in that year, a lot of grapes started turning red. But once they were supplemented, the new leaves that were growing out later in the season after harvest somewhat recovered. So, and also in our work, we noticed that the modulation of potassium transport is impeded under different water amounts as well. So this is an active area. I don't have a recommendation at this point.
There's a question on chat that came in for all of you. Can mealybugs transmit red blotch virus?
I can answer that. No. In one vineyard, a commercial vineyard close to Oakville Station, the red blotch spread was going in a crisscross where the leaf roll spread was also happening. So that's kind of evidence that the mealybugs were not transferring the red blotch virus.
I'd add to that the patterns of spread in the fields where we've monitored the spread are very different. With red blotch, it's a vector that's clearly a modal vector. So you see these individual vines starting to get hit at some distance away instead of seeing a more of a smooth spread across the field. And there's actually a paper from that we're on with the people, Von Walton and his group up at Oregon State University, where they've seen the same thing in Oregon, and where they have monitored the patterns of leaf roll and the patterns of red blotch, and they're very different patterns.
So this is kind of a question related to either the physiology of the vector. And, anecdotally, we saw in our own vineyard in Oakville, a lot of red blotch that coincided with a long period of drought. And it could have just been coincidental, certainly. But I'm wondering, would something like that, do you think that was coincidental, or does it have something to do with the climate and the vector, or the climate and the physiology of the vines, or no ideas at this point?
I don't think it's coincidental. In drought years, we see the disease express itself earlier in the season, very soon after raison. In years where we receive our normal allocation of rain, which is about 1,000 millimeters here in Oakville, we see the symptoms showing up very late in August, sometimes early in September. So plant water status and how the plant is able to move carbohydrates up and down is certainly interesting. and has an effect. And 1 year we tried to mimic the effects of red blotch virus just by girdling shoots and trunks, and we can see the same response from the plant if we girdle it by removing a piece of the bark from the shoot to the trunk, etc. Actually, Anita and I just have a paper published on that. Actually, got accepted today. So, but that did not have a red blotch positive plants, but it very much works the same manner of like, removing a, piece of the phloem in the case of these untoward weather conditions.
But there's a lot of other impacts too on the vineyards because, in wet years you have different resident vegetation present in the vineyards too. As well as the surrounding areas around the vineyards, and that could impact vectors in the vineyards and outside of the vineyards.
I have actually 3 more questions that came in on chat, so let me read those. Are there other plant hosts for this virus, or is it just grapevine?
Want to take that, Sudhi, or?
Oh, okay.
We
It's
both
okay, fine. You can take it, Frank, no problem. It was done by us together anyway, so yeah, take it.
You want me to do it?
Yeah.
Okay. No, actually, the only other hosts that we've found red blotch in, confirmed red blotch in, are wild grapes. And so it's probably limited to, Vitis species. And we've done quite a bit of survey work, and other labs have done quite a bit of survey work, and we have seen the presence of the virus fleetingly in other plants, but nothing where it's permanent in the other plant. Is that fair, Sudhi?
Oh yeah. Yes.
Well, yeah. Okay, let me go on to the next question. Have you considered looking at the interaction or importance of rootstock on expression and impacts of the virus?
I can take that question. We haven't I haven't been involved in the study, but there is a study funded by IAB, which was started by Dr. Adib Rohani, who retired from FPS a couple of years back. And Adib has been studying the rootstock and cyan interaction with respect to red blotch virus as well as other leaf roll viruses too. So the results are still, because I think it was started like in 2014. Some results are up there, but I don't know much about what has been found yet.
Yeah, I took over that study with Maher from FPS, so we will continue on that work. So there are 10 rootstocks in the study, and the cyan, the top portion of the plant, is a Cabernet Sauvignon clone 7, the most common cab clone we have. Again, in this case, we're seeing the modulation of net carbon assimilation being inhibited by different rootstocks, and this is going to be impeded by the hydric potential of the rootstock. So this work is going to go on for 3 more years to a final answer on the rootstock interaction. But the higher the photosynthetic capacity, or the secondary effect of our rootstock or indirect effect of our rootstock on the cyan, the better likelihood of not expressing the disease itself on the berries. That's what we see preliminary.
I mean, we did also at Oakville, the study site before we had to pull all the vines because we had no negatives left.
Right.
There was a complete randomized block design of 2 rootstocks in there when we did the Red Blotch study, and it was 110R and 420A, and we definitely did see an impact. The impact differing in impact depending on the rootstock, but the season also had a significant impact.
Right.
And to really dissociate those from each other can be very difficult if you don't have. We had 2 seasons. We wished we had more. But yes, there was definitely impact. 1 terroir, more vigor, 428, much less vigor, and you could see the impact.
So we have actually about now 4 more questions waiting, so we have lots of interest, which is great. Next question is anyone working with the lamp testing, and what are your thoughts on that as a useful tool moving forward?
Okay, I can answer that. LAMP is another test for the virus. Unless there is a way that it can be applied for testing for the virus right under field conditions, it doesn't have any advantage over the conventional PCR test or the qPCR test the labs are doing at this point. So that is, so right now, well, after Cornell developed that, actually prior to that, there was another test that was developed by IGEA. So these 2 tests are there, but they are not, to the best of my knowledge, they're not being used commercially. So it's an option, but yeah.
Great, thank you. Let's see, the next person is interested if anyone can comment as to the presence of red blotch in areas of the state with warmer winters versus cooler winters. For instance, Pope Valley, Lake County, much of Mendocino County seems less affected regionally than Valley Floor, Napa, and Sonoma.
Well, the vine is dormant during the winter. I don't know if you want to, I wonder if that has more impact on the potential vector than the plant itself.
Well, you know, everything is a little circumstantial. And we hear, I mean, we've had reports of red blotch from Southern California, you know, Temecula area, around one of the study vineyards that we've had for several years is in Santa Barbara County, near Santa Maria, and you know, the pretty warm areas. So it's hard to say prevalence, but I'd say that red blotch is in warmer areas and cooler areas, and even Pope Valley, one of our study sites is in Pope Valley. So we know that it's in that area, and at least in the Pope Valley site, there's pretty good evidence of spread. It's not one of the areas that we've actually mapped. So do you have anything else to add to that?
Yes, definitely. I think in 2011, soon after we found the virus and the Red Blotch Disease Association, everything, I've been trying to look at vineyards in different regions and cultivars. But by and large, I think it's mainly because its impact on Cabernet Sauvignon was much higher. So the Oakville area or Napa County, where Cabernet Sauvignon is the dominant cultivar grown, so that's where it was more. Even in the same vineyards, the grape wines tested in the previous year versus another year, the vintage-wise, it was different. So the effect was not always there, even in Napa County too. But if you compare, you know, Chardonnay versus Cabernet Sauvignon, I think it's not a good comparison. Because in Mendocino and part of Sonoma County, you have other cultivars dominate, you know, Pinot and Chardonnay, not so much Cabernet Sauvignon. But we have, I have seen, you know, covered people having effects in Paso Robles area and Fresno area and Foothills area. And it's not like a cool region versus hot region or anything like that. I think part of it is because how the vineyard pursued this problem and the cultivar that was being grown there.
So I guess following up on that, one of the other questions, and you kind of touched on this, Sudhi, is can you expect the severity of the disease, a difference in severity of the disease based on the length of the cycle of the cultivar? So I think, in other words, let's say you grew Chardonnay, Pinot Noir, Cabernet, and Merlot all in the same, exactly the same climate. Would there be a difference in the severity of the disease because some of those are early ripening versus late ripening?
Well, sorry. What are you trying to say? Somebody, okay, sorry. Okay, early on, I think my very first visit to a vineyard in 2008 was to look at some Syrah wines for a different disease. In the same vineyard, I happened to see Malbec having a red blotch-like disease at the time, but nobody knew. But I was told it was leaf roll. And a few years later, I confirmed that it was red blotch virus that was present in that, not leaf roll. The same symptom could be seen. This was in Sonoma County. The symptom could be seen in a Napa vineyard also. So the problem was by and large pursued by the Cabernet Sauvignon growers in Napa County, in Oakville in particular, but not in other places. I think it had much to do with the revenue losses that was being experienced by these premium wine producers in some regions.
Okay. Go ahead and keep sending in your questions or raise your hand if you have them. But we've talked a lot about the vineyard. I want to touch on the winery side. And let's say you're buying grapes or you're growing grapes that have red blotch in them. They're being affected. Anita, I wonder if you can touch on what, if anything, you can do in the winery to mitigate those effects.
Okay, so I'm going to go a little bit beyond the winery because most winemakers do actually have contact with the vineyards that they're buying their fruit from. We have been looking at the impact of just longer hang time as well. And in many instances, not always, by hanging the red blotch impacted fruits for longer, you can get to higher sugar levels, which does help mitigate the impact of the disease somewhat. What we found is when we originally only made red blotch positive and negative wines, harvested at the same time point, the impact on the sugar content is so great that you can add acid in the wine if you need it and you can equalize that out. But the impact of huge differences in alcohol content in the wine really overshadowed all the other differences when we did sensory descriptive analysis. Because alcohol has such a huge impact on the volatility of aroma compounds, on your mouthfeel, on the body of a wine. It enhances, it decreases astringency, it enhances bitterness. It has a multiple impact. So if you can get it to the same sugar level, or within like half a percentage, or even a percentage of ethanol to each other, then that impact is significantly decreased. Also, what we found was that you do also get an increase of the extraction of anthocyanins and other phenolics. It's not that you get much more anthocyanins synthesized or more phenolic synthesized, because a lot of that needs to happen earlier on with longer hang time, but you get better extractability. And we're currently looking into that. It could be that the delay in ripening is also delaying the natural breakdown of skin cell walls that helps the extractability of during winemaking, so that you can actually extract more of the phenolics that's in your grapes. We all know you don't get everything that's in there. So that seems to help as well. It doesn't totally mitigate the impact, but it definitely helps if you can do a longer hang time. We've also done studies where we looked at, okay, when is the impact so much that you should consider trying to do sequential harvesting, meaning not harvest the red blotch virus impacted fruit with the healthy fruit. The problem is because the season has such a great impact, you have to really check this out every season. And you could simply do it by just measuring the sugar differences, because it seems that everything else goes a little bit hand in hand with the impact on sugar. If the impact on sugar accumulation is much greater than other years, it seems to be the impact is greater on many of the other quality aspects as well. We don't have enough data to really say this for sure, but there's definitely a lot of circumstantial evidence for this. So looking at that, if there's a great impact, we saw in a year that there was an impact of 3 brix difference, that if you had more than 15% red blotch fruit included, you could see the impact, and mostly due to the alcohol differences. That has a huge influence. We have been looking at things like you're doing extended maceration, doing pectinolytic enzymes to try and get more of the phenolics that's in the red blotch impacted fruit out. It seems to be really good with helping you get more tannins out. At this time, we're not really getting more anthocyanins, but we've only done this for 1 season. So we're going to do this again. And like I'm saying, we're looking now also at the signs of actually what's happening at grape cell wall level. Are there compositional differences at similar ripening points between a grape berry from a grape red blotch impacted vine versus a healthy vine?
Great. Sudi, earlier you talked about roguing as being one of the main ways of dealing with the red blotch infected vineyard. I guess you could go on symptoms, but I was wondering if you could talk about, are there good ways of actually testing for whether your vine actually has the virus?
Well, there's only one way. There's no good way or bad way, unfortunately. So it's a lab test and a qPCR based test. That's the only way at this point.
And what part of the vine are you sampling to do that test?
Oh, okay. Yeah, we actually anytime after June, the virus presence can be tested. The younger shoots early on, soon after the bud break, a lot of them can test negative for the virus. But if if another 2 or 3 months are given after June, the leaf petioles can be tested. And then the dormant canes can be tested. And the trunks can be tested anytime, but I think that's not a good option to test anyway.
And I think related to that, with this testing, are there multiple strains of the virus that are out there? And does the testing cover all of those? How does that work?
The tests we have currently, they cover all the strains that we know. There are only 2, basically that kind of 2 different genotypes, but we don't have any biological differences between the 2, the genetic strains. So they are like 8% apart at the nucleotide level. So we call them strains based on the genetics, but biologically, we don't have any data or any differences that we know that these 2 strains behave differently.
Great. I just want to note at this point that I have another question, or maybe someone else from the group here has a question, but I do want to note that this office hours with David and Anita is a weekly thing, at least through the end of our academic year in middle of June. And next week, the guest on our show will be Dario Cantu, who's a professor in viticulture and enology, and his student Jerry Lynn. We're going to talk about a block that we've planted at Oakville that's a cross between Cabernet and Riesling, all different crosses between Cabernet and Riesling, that basically they can use to figure out the genetics of flavor development in grape. So I think it's going to be a very interesting show next week. I'd like to, not seeing another question, I'd like to end with kind of a question for all of you working on this project. This project goes through 2023, and I'm sure that you won't answer all the questions that you have now, and I'm sure you'll come up with more questions, but I'm wondering if Anita or others can talk a little bit about what you see as success when, by the time you get to 2023, what will you have accomplished that will make you think the project's been successful?
Well, I will take first stab, and then the rest can add on. I would say for each one of our main objectives, we just want to take steps forward. I think I don't want to lock Frank or Sudi and all the other principal investigators that's working on vectors into this. But yes, sure. I mean, it would be wonderful if we could say with 95 confidence that these are the vectors, these are the hosts, these are the reservoirs. But even if we don't get there, if we've made progress in, no, these aren't vectors, these aren't hosts, and these aren't reservoirs, but there may be some that we just haven't covered yet. I mean, that's still a lot of progress. So yes, I mean, we want to answer questions about what's the prevalence, how is it spreading, what are potential vectors, hosts, reservoirs, and hopefully Khan and I can, we will definitely make progress, even if it's just to tell you what does not work, right? Do not spend money or time on this. I can't with confidence say that we will be able to tell you how to fix this problem or what will work, but we will definitely have a laundry list of things that doesn't work because we are going to test them all. So I think that's great progress. And we hope to have more remote kind of sensing diagnostics tools, things that's cheaper and easier to do to cover huge vineyards. We definitely think we will make progress. I don't know if it will be completed by the end of this grant, and definitely have better numbers. We have Rachel Goodhue, that's also a PI on this grant, that's going to do economic analysis with us, working with us on the vineyards where we can show this is the impact. This is all the money you need to spend to maintain this vineyard. This is what roguing is going to cost you. This is what it's costing you to maintain this vineyard. This is the impact on grape and wine quality, and that people can make more informed decisions about whether roguing, if that still is the only option out there, makes economical sense for them or not.
Great. Well, that brings us to 3 o'clock. So first of all, I'd like to thank all of our panelists today for coming and talking to the group here. I know it's an important topic for a lot of you. I'd also like to thank all of you who came and listened in. And just a reminder, next week again, Dario Cantu and Jerry Lynn are going to come and talk about flavor development and understanding flavor development in grapes through their Cabernet by Riesling Cross out at Oakville, which I think is going to be a really important project. Dario's done a lot of the groundbreaking work in genotyping of grapevine and developing the tools to do that. So I think that's going to be a great program as well. So thank you again to all of you for attending. I'm going to turn off the recording. If somebody want to stay and talk to our panelists for a couple minutes, I'm sure that they would be able to answer questions for a couple more minutes. So
and if anybody had questions that we couldn't answer or didn't have time to answer, feel free to send us an email and we will respond by email.
Yep. So you can send email directly to Anita and she can get the question to the right person as well.
Absolutely. Thank you.
Thanks, everyone.
Thank you.